So here is the one that we ended up with, and we will start with this class 4.1. But first, let me ask is that, again, you have seen that I have not uploaded any new PowerPoint, and the one that I was talking about, I just uploaded that, or like replace the old file. Initially, it will look like the same, but some of the writings are a little bit different that you are going to see. So that's basically what it is. And so the first, let me ask that, do you guys have any question from the previous part that we talked about? Any questions on those part? Because the reason that I'm just have the little bit of luxury to do that, because we are not going to start a new chapter as such, because it's, as you said, long weekend and a difficult material a little bit. But any questions from this previous part so far? No? Okay, so if it is not, then we will start, and then you can always ask those questions for during the review as well. So that is all good. So what you're going to see, that one of the questions that you may be thinking, that why I am taking so much time on this chapter, the reason is that many of the things actually need a little bit of syncing time. We are talking about many things which you may not be that familiar with. And that's the reason that we are taking that much of time. What we will do that in the next chapter, we are going to look at the viral DNA transcription and the RNA processing that you have seen in the eukaryotic system. And we are not going to spend this much of time in there. We can just move through that within a week. So in the next week, we plan to do the 2 chapters as usual. But this is the reason that if not 2, at least 1.5, something like that. So that's the reason that we are taking some time. So let's start with this. So this is your, basically what we did the last week, and let me just go back to some of the questions that we have right here. Oops, sorry. All right, so we were in the Baltimore scheme right here, and we're just going to point out that where we were and what we did. So we did the reovirus, which is the double-stranded RNA virus that we have already seen. mRNAs are synthesized in the core particles. Now we are moving to the poliovirus and the coronavirus in the red. This is the new topic that we are just doing only for this year. I used to do the alpha virus, which is a similar but a little bit easier to understand a prototype of virus. With that, I replaced that alpha virus with this coronavirus this time. And that will be one of the topic of the honor students and the graduate students. They are going to dig into the details of the replication of the coronavirus. I'm going to give them a primer, what they need to look at and what will be the question. And then we have a little bit of more surprise that we will have just one, not much, because just one question from that discussion. The reason being that the honors and the graduate students are spending that much of time and putting them in there, and the rest of the class doesn't even read it. And that's the reason that, at least to pay the respect for their work, what they're doing, I will ask one question for that in the exam, and it will be not in between lines. If you read the discussion just one time, you will be able to answer that. I think that is fair to them. So you guys good with that, or war? You guys should be fine. Yeah, so you guys should be fine. No worries with that. It is just it is just more of just to have the respect that you all need to pay. That on top of this, they're doing this. So we will do that. And again, it will not be in between lines, very, very easy in that sense. So let's start with the next class 4, which will be the poliovirus. And you will see that the things will get a little bit complicated. You may need some extra time sometimes. And if you think that I need to just, and this is also one of the things that you can actually ask me, and I can do that. I can just do this replication on a complete whiteboard, and not in the class, but I can just record that and upload that if it makes it a little bit easier. That I can just do that if you think that before the exam that it is getting too much complicated as such. So those are all the options on the table. So being said that, let's start with that. So the plus-strand RNA virus and Flaviviridae family, Picornaviridae family are the members of that. And when you're talking about the flaviviruses, again, you do not have to remember these names, just it's a passing information that the dengue, Zika, yellow fever, West Nile, you hear all of these, and these are all under the flavivirus. And picornavirus example is the poliovirus. Pico small and poliovirus is very small, about 30 nanometer in diameter, and that's why they named that family as Picornaviridae, and the poliovirus is the name of the genus. So what you have inside that, you have, in general, you do have a plus-strand genome RNA. Now remember that here when we're looking at this plus-strand genome RNA, you are seeing that there is a 5' cap, and this is needed for the ribosome to understand that this is an mRNA. It needs to be capped. And then you also have a poly-A tail. AN means N could be 150, 200. So 150, 200 A's are added to these, and this is what we call as a poly-A tail at the 3' end. So this is a ribosome-ready plus-strand. Now, all of the viruses in this family doesn't come with exactly the same. There will be a little bit of difference in between them. This RNA needed to be processed to some extent, and we are going to talk about that in a different chapter. But just for the overview of that, pretty much all of them are kind of the same thing. So think about that. It's a plus-stranded RNA and almost ribosome ready. So in the first step, what you are trying to do is that, of course, you will make from this will be translated directly, and after translation, you make the proteins and the RNA-dependent RNA polymerase, because the plus-strand RNA viruses do not carry their RNA-dependent RNA polymerase in the particle. So once this mRNA is translated, the RNA-dependent RNA polymerase and the other proteins will be manufactured in the cell. Those will carry out the replication. So once this is done, then from the 3' end, remember that, it will start synthesizing the 5' to 3' end. So this is plus strand. This mRNA will be used as a template, and those RDRPs will replicate that from this end, 3' end complementary, and the complementary will be 5', and it will synthesize in this direction. So look at this arrow. The arrow means is the direction of the synthesis. So this is very, very important. This is a subtle thing to understand that in which direction the synthesis is occurring. So from this end, this will be the template stand, and this will be the strand which will be complementary to that, and it will produce this minus strand full-length complement. And this is transient. This will be degraded, and this has no other functions. The only function is that to use this one as a template now. And on this template, the RDRP will the same RDRP. There is some modification like a protein addition or something like that. You are going to see that. It's going to synthesize the mRNAs once again, or the plus-strand RNA, which is the genome. Now, this RNA, when synthesized, can be used as two functions. One, it can be used as the genome to package inside the virion or the virus particle, or this can be used as more of these mRNAs, because see that one virus gets into the cell, that will do this. But that virus does have one genome. If there are two viruses, just only two genome, one or two mRNAs. You need to produce many proteins, and these are limited by just one or two genome mRNAs, and that is not sufficient for the productive viral synthesis or replication or the reproductive cycle. So you need to make more mRNAs, and that will be synthesized on this template, and some of them will be packaged into the virion particle. Now, this is the very generalized scheme. What you are going to see that many of the viruses will not make this full-length mRNA out of that. Instead, they are going to make shorter than full-length mRNA. Again, I'm repeating, shorter than the full length of the genome-length mRNA, and we call them as a subgenomic mRNA. I'm again repeating, some of the viruses will not make the genome length means this genome length mRNA, but they will make shorter mRNAs out of that, which will make some of the other proteins. And there are many other properties that they have, and it varies. So it is very difficult. So the scheme overall is the same, but there are many intricate differences in between the viral families. But the basic understanding is that from this plus sense 5' to 3', it will make a negative sense 3' to 5'. And then from this negative sense, it's going to get back the plus strand or the plus sense, which is the mRNA, which could be the full length, this size, or it could be smaller than that, which we call as a subgenomic mRNAs. And one of the things that I also included in there, so the plus strand right here is recognized by the cell as an mRNA. So that is not going to be attacked by the RNases which you have in the cytoplasm. But what happens that as soon as you synthesize this minus strand complement right here, this is unusual. You do not have anything similar to this in the cell, and the RNases are going to attack this. So there are some strategies that the viruses do use to protect these RNAs. There are two strategies. One of them is that they coat this minus strand with a protein or block the ends with a protein. Sometimes they actually do put them on top of a membrane vesicle, and these membrane vesicles are induced or produced by the viruses as well, which actually make an enclosure or a covering around these around the RNA molecule so that the RNAs cannot access these. And this is how they temporarily protect these RNA molecule, which is only the minus strand. The plus strand doesn't need anything because you have a cap structure, and you cannot digest this because this is exactly similar to the mRNAs in the cell. All right. So the next, what we are going into, that we are going into the reproductive cycle. And first, we are just going to only talk about the ones which is needed for the RNA-directed RNA synthesis. So once the virus goes into the cell, we talked about that, it releases the genome, and see that there is a yellow, the orange dot at the 5' end, and we're going to talk about that. It is a protein covalently linked with the genome. In case of poliovirus, it's called a protein called VPG that's attached to that. That's very specific. So now from this, the ribosome is going to actually make right here from this end, it's going to try to translate. Now, although you can see right here, this is actually I although wrote poliovirus, but this is actually all of the Picornaviridae family. And although you are seeing that there is a 5' cap that is being translated by the ribosome for the poliovirus, you actually do not have a 5' cap. You have a similar structure, which not similar structure, you have a different structure at the 5' end, and that is recognized by the ribosome to synthesize that. So this 5' cap is in the Picornaviridae family. Probably, I'm just going to need to just probably change this a little bit. Picornaviridae family, but exception with the poliovirus in there. But anyway, the overview of that is that it's going to get translated, and the translation of the whole genome one into the other, except a few bases at the ends, a long single protein is formed. This is called a polyprotein. So this is like a one polypeptide chain attached to the next, to the next, to the next, and it's a large one single protein. So what's going to happen that this long, large polyprotein that is produced from this mRNA is going to be degraded by the proteases, and these proteases are the part of the polyprotein, and they do have an autocatalytic function. That means even if they are in the part of this protein, as it folds, those proteases, just by folding on top of them by itself, can cleave this long polyprotein into individual protein. And this is just the first set of cleavage, which is the P1, P2, P3, and it will be again again cleaved into much smaller ones, and the whole thing is not being shown here. It is a little bit more elaborate. But just to know that the overall from here for this slide, that from this polyprotein, you just make lots of proteins which are the RNA-dependent RNA polymerases, as well as the structural proteins. So once this is done, along with this, what is going to happen, that the mRNA, which actually comes in, which is the genome, it is getting translated. But along with that, some of the mRNAs that is in the cell, they are going to associate themselves with some of the membrane vesicles. And on top of these membrane vesicles, you are going to see that this is a double-stranded mRNA, that is the double-stranded RNA that has been formed. And you can see right here, you have the negative sense, which is the olive right here, the negative sense that has been synthesized on top of these vesicles. Once this is done, you can see that that negative sense can be used to make more of these mRNAs. So those can be used. You can see that right here, from this double strand, more of these mRNAs can be produced that will produce more of these protein, and the cycle will go on. So this is the brief overview of the plus strand RNA viruses. And again, poliovirus is a standard example of this, but it does do something different depending upon what it is. So let's take a look at how the genome of the poliovirus looks like. So here is the genome from 5' to 3' end, and here is a protein called VPG covalently linked to the 5' end. And then you have an untranslated region right here, and then you have an untranslated region in the 3' end, followed by a poly A tail. Now, you can see that there is no 5' cap in here, and this protein is not a cap either. So we are going to explain that how translation actually happens. You are going to see that in this untranslated region, there is an RNA secondary structure forms, which we call as a cloverleaf structure. And this cloverleaf actually does allow the ribosome to enter in the middle of this RNA. And we call this one as an internal ribosome entry site, or IRES. And we're going to see that in the next one. So here, let's take a look at what are the products which is formed from the mRNA, or the genome length mRNA. So think about that. This is the whole length. And from here, you are going to synthesize, the virus is going to translate these, not the virus, the cellular translational machinery is going to translate this mRNA into the P1, P2, and P3. And you can see that in them, you have some specific areas, like right here. This 2A is means it is a protease. And right here, you can see that there is another protein called 3C. That's also a protease, 3C pro. So on the other hand, you can see that there's a black point right here, which cleaves this VP0 into VP4, VP2. This is not really known, which exactly does this, this proteolytic cleavage. So what's going to happen, that from this P1, P2, P3, by this autocatalysis, it's going to make first these set of proteins. So VP0, VP3, VP1, then 2A, 2B, 2C, and P3. And again, all of these names are not necessary for here. I will just specifically tell that which are the ones which is specifically needed. So then what happened, the 3C Pro, you can see that first the cleavage of this P1 and P2, P3 is done by this 2A Pro. And after that, that 3C Pro actually cleaves autocatalytically all these different areas. So from the VP0, you get the VP2 and the VP4. So the viral capsid, just if you remember from the previous one, we talked about that, VP1, VP2, VP3, VP4. These are the structural proteins which make up the capsid. On the other hand, these are the proteins which are responsible for the proteases and RNA synthesis. And here you are seeing that the P3 is to be cleaved into 3AB and a 3CD Pro. 3CD Pro is also a protease. And then here you have a 3A, 3B, and this 3B is the VPG that is right here. And the 3CD Pro is going to get cleaved into the 3C Pro, which is the protease, and the 3D Pol. Now you know that why the RDRP of the poliovirus is known as the 3D Pol. So this 3CD Pro is divided into C and D, 3C and 3D, and this 3D part is the polymerase. And this is now going to synthesize the rest of the RNAs, genome RNA and the mRNA in the cell. So next, what we are going to see that how the poliovirus actually have this internal ribosomal entry site. So the poliovirus 5' end of uncapped mRNA. So what happens, your VPG is directly attached with a phosphodiester linkage to the first residue of that mRNA. So right here you have the, so what is this base? Anyone who is good at biochemistry? Anyone? Can you tell what base is this? No? Guess? Wrong is fine. No, so this is a U. So this is uridine. If why it's not an adenine, you will see, or I'm not adenine. That would be a purine. So this is a pyrimidine, and then if you have a CH3 at the 5 position, which is 1, 2, 3, 4, and 5. Here, if you have a CH3, it will be a T, which is a thymine, which is present in the DNA. But if you don't have the methyl group here, CH3 group, this is a uridine. So the first residue is U. The second residue is U as well. So the first residue, U, is covalently bonded with this phosphodiester linkage with the VPG, and this will be cleaved by a protease in the cell. So now you have an uncapped plus strand RNA, but how ribosome is going to understand that this is an mRNA? You don't have any cap structure. So but what happens that right here, a particular cloverleaf structure like this, a secondary structure is made, and this is understood by the ribosome, and the ribosome actually can enter, which is called a IRES, or internal ribosomal entry site. And precisely on the base 741, from the 5' end, the base number 741 is the start site of the translation. So this is basically what happens, and all you have to know that how without a cap the poliovirus RNA is translated by the ribosome. It is done by the IRES, or the internal ribosomal entry site. So the next thing that we are looking at some of the structural element, and this is not at a particular site, and we are going to see that what are in a particular site and what not. So if you think that this is the 5' end, this is the 3' end, and the 5' end right here, you can see the VPG, and right here is the clover leaf that we just saw in the previous slide. It's just upside down here. So that's the clover leaf, and this contains the IRES, or the internal ribosomal entry site. Then you have another feature on the RNA, and this is what we call as a Cre element. This is a cis-acting replication element, and this is also responsible for the binding of the 3D Pol, or the RNA polymerase. Interestingly, what happened, this CRE, although it is shown in the middle of the 3', 5', and the 3', this is movable. This can move in different areas, and it can form this. This can be a CRE as well. This can act as a CRE. So this is just a feature. It's just been shown here as a schematic, but think about that. This can be this whole clover leaf. This can act as an iris, but in them, this could be a cis-acting replication element as well. So it can move here and there. On the other hand, the 3' end also do have a structure which we call as a pseudoknot, and we are going to see that how this helps in the replication of the poliovirus. So the interesting part is that there is a specific way that the control of the ribosome is done by the viral RNAs, that the cellular polyislated RNAs are not copied by this process. So the RNA-dependent RNA polymerase of the virion or virion-coded or the virus-coded actually understand that it will not copy any of these, because you will see that although they don't have a 5' cap, the cellular mRNA is also plus-stranded. And just to make sure that the RNA-dependent RNA polymerase don't actually copy them instead of the viral RNA, there are some specific structure, and these are some of these. Now, why it is called a cis-acting? Because if you just see that if these structures are on the same side of the RNA as a secondary structure, then we call them as cis. But if this structure is formed on the other side, we call it as a trans-acting element. And if it is just like going on, like if it just rounded up and it is forming like this. And we are going to mainly talk about the cis-trans elements in the transcription chapter. Here it is not important. Just remember that what are the different features. So now let's take a roll. We are just only looking at just one of the CRE and this CRE element, that how it actually helps in starting the replication. So first, what happened in the CRE, there is a 3CD, and this is your 3CD Pro. So remember that this is the 3C, and this is the 3D, but when it is 3CD, it is a different protein. And two molecules of them actually bind to this stem loop structure just into the loop. And this actually helps to melt the loop, and this allows the recruitment of the 3D. Now see that this 3D CD has a different function. Here it is not self-cleaving them into the 3CD, is not self-cleaving into the 3D and 3C. In the other than these molecules, there are 3 CD molecules free in the cellular environment or near the membranes. And the 3C is going to cleave them into the 3D, and this is a free RNA-dependent RNA polymerase in the cell. The melting part of the CRE element is done by the 3CD, the whole of 2 molecules of the 3CD, particularly the 3C dimer part of the 3CD. So again, repeating, the whole thing is 3CD, and the melting of this actually is achieved by the dimer of the 3C. Once this part is melted, then what happens? It recruits one of the 3D. This is the RNA-dependent RNA polymerase. And also, this 3CD will do something that we are going to see. This is going to actually process and bring the VPG by cleaving that from another protein called 3AB. And this VPG will now attach onto the specific residues on top of that, which will be added with 2 uridines. And this is a de novo synthesis. You can see that it is a protein, and it will be directly added with PU, phosphodiester linkage, uridine, phosphodiester linkage, uridine. And this will act as a primer for the RNA synthesis. So this is the first thing that what is happening. So what is the basis of this? Remember that this VPG is added onto this right here, onto the polymerase. Now, where this U comes from? So on top of this loop, you will have the complementary 2 A's. So on top of these 2 A's right here on the loop, these U's are the complementary to that. That is being synthesized by the RNA polymerase and attached covalently to that. So that's basically the overall. So there is no primer or anything like that. You just finds the base right here, A, and puts an U here. The next A, it puts an U here, and it's covalently linked. So this will now act as a primer for further RNA synthesis. So here, just make sure I just try to write it as much detail as possible, that the whole process, that how it is happening. And this is the synthesis of the intermediate minus strand. Understanding the plus strand is easy. The minus strand, which is the intermediate, is a little bit difficult. So let's take a look at how this happens. So right here, what you are seeing, that this is one of the part of the membrane vesicles that are produced in the cell that we saw as those round structures right here. These are the membrane vesicles, And that's one of the part of, that's one of the part. So what you're seeing that right here is the protein 3AB that we were talking about. And this is a fusion protein. And you can see that orange part right here, which is sticking out, that part is the VPG. And see that there is a hydrophobic residues right here, which is buried into the phospholipid membrane. So this is a membrane. Now you have this part where you are seeing the VPG and this cloverleaf structure, CRE element, another CRE element, and the pseudoknot. This structure is the viral genome RNA, 5' to 3' viral genome RNA that comes in. So this actually binds to some specific things, and how it happens that it binds to a protein called PCBP, or a poly-C binding protein. Where the C's are, these C's are on this CRE, or the cis-acting element of the clover leaf. So the whole structure is a clover leaf, but each one of them has a CRE. So each one of them. So this is binding to this PCBP in right here. And the PCBP interacts with this VPG as well, right here. VPG part of the 3AB. On the other side, on this CRE, you can see that here the 3CD Pro is attached. So this is your 3C and this is your 3D. Now, you can see that in the previous picture, you have two molecules right here, and that is needed. Here, just for the simplicity, just only one of them is shown, but actually there will be two of them which will melt this part for the recruitment. So now what is happening that this 3CD Pro does two different things. This will actually attach to another protein called PABP, poly-A binding protein, right here. This is one of the function. Another, this 3CD Pro, and again, remember that this is a model. This is very close to this, and the 3CD Pro will cut or chop the 3C, will chop this, and a VPG will be cleaved out of the 3AB. And this VPG will be the one which will be used by a free 3D Pol, which will act as a primer. So let's go one at a time once again. This is being associated with the poly-C binding protein. Opposite to that, you have the 3CD Pro. 3CD Pro binding to a poly-A binding protein that attaches to the poly-A tail to the 3' end of the genome. So here, one take-home message. This is what is actually the most important part for your exam or the quizzes that we ask, that if you just realize that right here is the 5' clover leaf, and the 3' end is brought almost close to the 5' end by the interaction of this poly-A binding protein and the poly-A tail. And we call this as a circularization of the genome RNA. And this is one of the key steps that in order to produce the minus strand, it need to actually completely circularize the plus strand. On top of that, the genome will be synthesized. So that is the first step. Now, once this circularization is done, you saw that this 3C part of the 3CD Pro will cleave that VPG. Now, this VPG, which is not shown, this VPG will attach onto another molecule, which is not shown, and it will put the two U's on top of that, which will be this phenomena on another free RNA molecule, which is not attached to this. So this is one of the template. It will use to synthesize this one with a free, like this, another template. So once that is done, this VPG PU will be transferred to this poly-A tail right here. And this specifically, this transfer, how this transfer is actually done is not clearly known, but this is basically the overall process. And now what happens using this primer, the polymerase will go from this one end all the way down to the other end. So it will just basically go through this all the way to the other end to synthesize the 3' to the 5' end. So that's basically the overview of the whole process. And it may sound a little bit difficult in there. Try to read and draw it, and it will make it a little bit easier. So now you have the 3' and the 5' end, and this is the temporary or the transient negative strand. On top of that, you will synthesize the plus strand by the same process, basically. So it is just the same. So that's the overview of the poliovirus minor strand synthesis. So now we are looking at the coronavirus. Let's see before we ask. Any questions from there? You may have a lot of questions, but just if you just have any questions for quick idea, many of you is like, I just did not understand that. That may be the feeling, a little bit overwhelming. But if you have any specific question, I can answer. Anyone? Good? Okay. All right. Now, next, let's go to the coronavirus. And this is a self-explanatory. These are all the different proteins that you have in here. This famous spike protein is there. It's a trimer, and because it's a fusion protein. So you know that this is your class 1 fusion protein. And here you have the huge genome, which could be in between 28 to 30, 32, 35 kilobase. There are some which close to 40 kilobase, largest known RNA genome plus strand. And they are encapsulated with a protein. So that's a unique thing, whereas the plus strand of the poliovirus is not encapsulated. So how this actually happens, and it is a very complicated process, and that's why it is not usually used as a standard plus strand RNA virus synthesis. So once the virus actually gets through the receptor-mediated endocytosis, you all know the spike protein ACE inhibitor, ACE2 inhibitor actually gets it by the endocytosis, and then the long mRNA that comes in, that will be translated by the ribosome, but not fully. It will make two different things. It will make a 1A, and then it will make a 1AB. So how this is possible and how ribosome does that 1A and 1AB. And the way it does that, at the end of 1A, there are some very specific A residues. On top of that, the ribosome actually slips, and it slips out of the frame, and the coded proteins actually help that, and the ribosome cannot correct that. So it slips out of frame, and what happens that usually, this is again, we should not go into that much detail, but remember that for the eukaryotic translation, each mRNA has one long open reading frame. That means that you start with an AUG, you just go all the way to the end, reading 3 bases at a 3 codons at a time. And then when you have at the end UGA, UAG, or UAA, any of the 3 stop codons, then at that point, your translation actually stops. Now, what happens on this particular event, the ribosome starts making the first ORF, or is reading 3 bases at a time. In one of those A residues at certain junctions on the gene, it slips out of the frame. And when it slips out of the frame, it will not read. And this is just like you think about your frame shift mutation, that kind. It will slip out of the frame, and then it will keep going again, making this bigger protein. So what happens, this 1A and this 1AB, although it is written as 1B, and we call this as 1AB. Now, remember that this book from where I just took these pictures, this is 2015. A lot of material has been added after that, and I had to modify some of these pictures as well. So these proteins are going to make all the non-structural protein which are required for the synthesis of the mRNA, including the RDRP. So the RDRP, RNA-dependent RNA polymerase, are made from this protein. Now what happens that you have these double membrane vesicles. These are also used by the coronavirus for their synthesis of the genome and the mRNA. So from here, what you're going to see that the virus is not going to make the intermediate short size subgenomic negative sense RNAs, On top of that, it's going to make these subgenomic mRNAs. These mRNAs are much, much shorter than the genome length mRNA, and these are going to make these proteins, which are the structural proteins. Remember that non-structural proteins like RDRP and the regulatory proteins comes from the direct translation of the genome. Once the genome translation is make the RDRP, it make the switch that it synthesize the intermediate and the subgenomic mRNAs to make the structural protein, and also it replicates the full length negative sense. On top of that, it replicates or synthesizes the full length genome, which is packaged inside the virus, and the virus actually assembled into the endoplasmic reticulum Golgi intermediate complex that we are going to see in somewhere else, and it exits out. So how this is actually happening, and this is one of the things that not this weekend, but it is due next weekend, the graduate students and the honor students are going to go into the detail of this one. So here what we are seeing, that right here you have the 5' end to the 3' end of the coronavirus genome. And here you have the 1A and 1B. So these 1A and 1B is out of frame. So first, once this goes in, it first going to make the 1A protein, is going to shift, then synthesize the 1B, which is attached to the 1A, and it's going to make 1B. And this is your RNA polymerase. And now you can see that right here you have 1, 2, 3, 4, 5, 6, 7, 8, these proteins. Now remember that, don't think that this makes one protein, this makes one protein. One AB protein usually makes almost 18 or 19 different proteins. Altogether, the coronavirus actually do produce about 27 proteins. So 8 of them are actually have structural implications. On the other hand, the rest are regulatory and RNA-dependent RNA polymerases, and some of them do interact with the host immune system as well. So once this is done, then the mRNAs will be produced, and you can see that these are the mRNAs which are of subgenomic length. But the synthesis of the mRNAs is very complicated in the sense it is very, very unique and not seen in any other, like most of the other viruses actually don't do that. There's some other example, which is called a nested 3' co-priming. Now, let's look at one point at a time that what we are looking at. So here, right here, I had to put all these marks in here so that it makes actually clear. So now remember that this I just did not have enough time to label these properly. Remember that all of these purple that you are seeing, all of these are TRS, or these are called the translational regulatory sequences, TRS. And here you have a leader sequence. So what we are looking at is this red label right here. This is the genome plus 5' to 3'. So this is the way the genome is. And you can see that the genome has a 5' leader. Then you have a TRS. Here you have a TRS at the end of right here. Then you have a TRS and lots of TRS at the end of each of these protein junctions where it is coded with that. And I just tried to this was not in the book, I just tried to make those TRSs. Now, when I am trying to make the mRNA, that very carefully try to understand that, you have the 5' to 3' end, and the RNA polymerase will start at the precise 3' end. And we are not going to the top. We will talk about that later. So it will start, so it is starting right here at the 3' end, and it is traveling. It is going through this TRS, going through this TRS, and going through this TRS, and it decides right here, from this TRS, it's going to jump all the way down to this TRS and synthesize the rest of it. So now what happens, that you have synthesized the mRNA, which is attached to this part and this part. This whole portion is looping out, or this whole portion is looped out of the RNA polymerase. It just jumps from this TRS to this TRS. So where is this? This is there. This is not that it is chopped off, but it is just looped out in there, and this is the process. So you have this one subgenomic mRNA is formed. So think about that. If that happens, you get this mRNA, where you have all these genes on this mRNA. Now, the interesting thing, what happens, now think about that this N is the last one right here, is the last one. So how you synthesize this one? So to synthesize this one, you start from this end right here, you go to the first TRS, and from here, you will jump all the way to the leader and just synthesize this leader. Remember that the leader sequence is not your translation product. So it will jump from here to all the way to the here, and it will have that. So if you have the M and the N, and if you just think that M and N are the last two, the synthesis will start from here. It will go up to the second TRS. From here, it will jump all the way up to the end. So at each of the TRS, the ribosome actually can jump, and they can make all these different nested mRNAs with this. So now your question is that what is the purpose of this? You have all of these attached end to end, but remember that at the end of each, you have a translational regulatory sequence. So ribosome will stop synthesizing the rest of the products. So you can see that right here, although the mRNA contains all the different proteins, it is shown as 2A. That means the mRNA is going to be translated to 2A and stops. 2A is produced. It's going to translate HE, stops. HE is produced. And this one just only have the N, and it would only produce the N. And this is basically how the translation actually happens. So really complicated, and I will give you guys, the honors and the graduate students, some of the video links or some of the papers from where they can start, and they will be able to write this down nicely, I'm sure, and I can just intervene as well. All right, so the next one is the negative strand RNA viruses, where is the vesicular stomatitis virus, and is an example of this. And you can see that here you have the 3' to 5'. This is the negative sense genome, and from here there are two things that is happening. Oh, one thing I just forgot. Just to mention right here. So we talked about the genome length, but remember that the virus also do have the ability to start at this end, the top, and it can actually synthesize without making this jump. It can also read all the way to the 3' end and make the genome length mRNAs as well, and that will be packaged into the virus. So from this, it can make the intermediate with these nested deletions or omissions. Also, it can make the full length complementary of that. And once it makes the full length complementary genome, full length complementary minus genome RNA, on top of this, you can make the whole full length of the genome RNA as well. So that's the last part that I just wanted to mention. So here, on the other hand, you can see that you have the negative sense 3' to 5'. And from this 3' to 5' negative sense, you can get mRNA synthesis 5' to 3'. And also the mRNA, which is the intermediate from this, this is used as the complement of this, on top of which you synthesize the 5' to 3' in this direction as well. So this is basically the overview of that. And the vesicular stomatitis virus does have an unimolecular genome. This is only 11 kilobase long, just only one long genome itself, not segmented. So what happens that you can see that once it goes in, from this long single genome RNA is going to synthesize several different proteins. And the matrix protein, P protein, phosphoprotein, it's a nucleocapsid protein, and the L is the RNA-dependent RNA polymerase. And there is also another protein which is associated with the ER. This is the glycoprotein. And why it is associated with the ER? Because this has the hydrophobic sequences, and this is actually inserted into the membrane. That's why this is synthesized on the ER-bound ribosomes. So these actually are going to make the intermediate plus strand, and also the mRNAs. Remember that from here, it also will make the mRNAs, and also make the full genome length RNA to be packaged into the virion. So here we are looking at the genome organization itself. You can see that right here, you have the 3' to 5' end, and from here, you can synthesize a leader, which is not capped. You can synthesize all these different proteins, N, P, M, G, and L, large protein. This is the RDRP. These are all capped, and you have a 3' poly A tail. So what is the function of this? That we are just going to come to that in a second. So let's take a look at how actually mRNA synthesis happens. So you can see right here, it is a portion of the genome that is making these mRNAs, very different. And this is actually done by this process, which is called a stop-start model. So in this process, what happens, this RNA-dependent RNA polymerase start at the precise, this intergenic sequence in between the leader and the end gene at this intergenic sequences, and it will just go all the way to this intergenic region, and it will stutter there. The reason it stutters, and we're going to see that, it contains a tract of 7 U's. We call them a poly U7 tract. It will stutter there, and that will actually make lots of these U's by slipping at that point. And some of the ribosomes will fall off. Some of the RNA-dependent, not ribosomes, the RNA-dependent RNA polymerase is going to fall off. Some of them is going to continue and make some of the P's. Then some of them is going to continue through the M and the L. So what happens, it starts with the N, but at each intergenic region, as the RNA-dependent RNA polymerase slips, less and less ribosome, I just keep saying ribosome, less and less RNA-dependent RNA polymerase is going to go through each of the intergenic sequence. So there is an attenuation of the mRNA synthesis. In maximum number of mRNA, little less, L RNA-dependent RNA polymerase is produced the least in amount. So this is a nice way to control the production of the relative amount of the mRNAs. So what you are seeing that here with the RNA-dependent RNA polymerase, some of these are actually associated with that, some of the phosphoproteins, and these are some of the host factors. And these are some of the things that work I have done in 2000, like published in 2004, that I just put in the, you can just take a look at that. And this is beyond the scope of this course, but these are also involved in the function of the RNA-dependent RNA polymerase of the VSV. So again, so what happens that at each intergenic sequence, the RNA-dependent RNA polymerase stops and again starts, stops and again starts. And this is basically the stop-start model of the mRNA synthesis. Now here it shows that how the poly A is done. This is one of the intergenic sequence, and you can see that you have these 7 U's in there. On top of this, the RNA polymerase slips, and it makes out this, as it is sleeping, it's just on this, it is just trying to go back and forth, and it's slipping, and it pumps out this 150, 200 A residues. So each mRNA will have the poly A attached to that. And then you will start the next gene. And the next gene is always starts with the very specific sequence. You can write here, you can see that UUGU, you can see that right here, UGU, this is the start of the next one, right here, ACA. This is the same for each intergenic sequence in there. And here it is capped by a 7-methylguanosine. You can see that. And this is the start site for the next genomic sequence. And this is how at each intergenic region, the poly A tails are added. So now what happens that if you remember that the RNA polymerase also do produce a little bit of these leader RNAs, which are not capped. And what happens to this? We have just only, when we're talking about the mRNA, we talked about that it starts from here and it is capped, but it also makes a little bit of leader, which is uncapped. See that what happens that once all these proteins that is made from this, right, all these proteins are in sufficient amount, particularly N, remember that the N protein is produced the largest in the amount. Once those proteins are available, as soon as this leader is actually formed, right here. So this is your genome right here, 3' to 5', and the leader is formed. Once there is enough amount of N protein, it will immediately encapsulate the N protein. And the RNA polymerase, along with the N right here, see that RNA polymerase here, which does the mRNA synthesis, the no N is associated with that. Now the N is associated with that, and this actually gives the polymerase a different function. And we used to call them, and it was also in this paper that you can see, that we call this complex which makes the mRNA as a transcriptase, because it just basically, the old term, transcribe the mRNAs, whereas in the presence of N with a little bit of different association of the host factors, it makes a replicase which makes this replication. So these two RNA polymerases are different in nature. Basic structure is the same, but the hollow enzyme, which is associated with other factors, are different. So what is happening that the leader is made and it is covered by the N protein. As soon as it's done, now this on top of this, it can synthesize the rest of it. But what will happen that when the N protein coats the whole of the minus sense, that is the strand that is being synthesized as it is coating, it cannot slip from the intergenic region. Again, remember that what I'm trying to say. The previous one, what we were seeing, that when you are synthesizing this mRNA is naked. It is not covered. In this case, it starts from here, and as it is synthesizing continuously, as this is being synthesized, it is continuously being enwrapped or encapsulated, and the intergenic region is also encapsulated. And at that point, this form of the RNA polymerase cannot slip, and it will go straight through the intergenic region all the way to the end, and it will synthesize the full length of the intermediate plus strand RNA. And from that, again, it will synthesize the same way all the way to the 5' to 3' end of the negative sense genome. And again, as it's synthesized from the 3' end, it will keep encapsulating that with the N protein, and you get the full length complement of that. This will be your genome RNA. So that is the switch that initially the mRNA synthesizes the protein required. When the protein, particularly the N or the nucleocapsid protein amount goes high, at that time, the mRNA synthesis switches to genome replication or genome synthesis. So that N protein is there are some host factors involved, but that's basically the overall process. Now, the next one is the influenza virus, and this is exactly the same slide as the VSV, so I'm just not going to explain that. And here, it is a complicated process, but the only thing that is happening here is that it happens in a nucleus. So this big box is the nucleus, and the nucleocapsid is directly delivered into the nucleus. And from there, you can see that the mRNAs are made, and these mRNAs are transported into the cytoplasm, which makes all these different proteins. And out of these proteins, you will going to see that PB1, PB2, and PA, these yellow ones, are the RNA polymerase. And they are going to move inside the nucleus, transported inside the nucleus by a nuclear transport signal. And they are going to synthesize the plus strand intermediate. From the plus strand intermediate, it's going to make the minus strand. And also the plus strand intermediate is going to also make the lot of copies of the mRNAs as well. So you have 8 different segments, just one shown here. But remember that all of these 8 different segments are going to do the same thing. So here, if we just look at the structure, these are the different segments inside, and each one of them is attached with this PB1, PB2, PA, which is the RNA polymerase. And these are the three of them. So they are coded in segments 1, then they're coded in segment 2 and segment 3. And then you can see that these segments actually make the other proteins, which are mainly the structural protein. And some of them, by the splicing, you can see that they can one segment can actually make two different proteins, M1, M2, NS1, and NEP, NS2. So the number of proteins are here also more than the number of mRNAs that you can actually see. This is by what you call as an alternative splicing that we are going to see that in the next chapter. So one of the very interesting thing, this whole thing of this individual is that very similar to the VSV. There is no difference in that. One of the important thing is that how you make the caps. In the VSV, the RNA-dependent RNA polymerase just makes the cap because it does have the 7-methylguanosine activity. Here, it is very different. What it does, that if you just see right here, it shows host mRNA. Why it shows host mRNA, where it comes from. This host mRNA is actually needed because this is the most interesting part. The RNA-dependent RNA polymerase cleaves this cap right here, cleaves this cap, and this cap is now attached to the end of these mRNAs. So what you are going to see that right here, this portion comes from the cap of a host mRNA, which is cleaved and snatched, basically stealing the cap of a host mRNA, and that mRNA will be completely non-functional. And using this, it's going to attach it to the second penultimate base. The first one is U, second one is C. It's going to attach to the second penultimate base, and this will act as the primer, and the synthesis will go on. So snatch portion of the 5' cap, and this is one of the unique thing of the influenza virus. On the other hand, how this is being synthesized, the mRNA is synthesized by a moving template model. So what happens that the 5' end of this intermediate, you can see this is the intermediate, is attached to the polymerase, and as this threaded through this catalytic site, the plus strand is being synthesized. So think about that, that 3' initially was here, and this actually is pulled through like a ratchet-like motion all the way through. As this is being pulled through, which is a moving, this is the template, and this is the moving template, as this is moving through this side, this mRNA or the genome RNA, whatever the case may be, is formed. So now what happens, it goes all the way to the end, and this U residues, these 7 residues of U again comes back into this active site. And right here, as the 5' end of the template is attached right here, it cannot pull farther. So here the RNA polymerase again slips and make a lot of A's out of just these 7 U's, and the poly-A tail is being made. So that's how you add the poly-A tail. And this is exactly the similar picture of the VSV. You can easily see that as soon as this 5' capped, the 5' unprimed RNA is capped with this NP protein, instead of the N protein, this is the NP protein. So that's the only difference in between them. So the difference, what we have seen, that here you have a host M7, which is the methylguanosine 7 primer, that is different from the VSV that is in there. On the other hand, there is no leader that is synthesized in here. It starts right from this sequence, and when that portion is covered by the nucleocapsid protein, you can see that it will, as it goes on, it will get encapsulated, and you get back the 5' to 3' end. This will be again used as the intermediate to synthesize the unprimed genome RNA, which is this. So the only difference in between that in the mRNA synthesis step, the RNA-dependent RNA polymerase steals the host cap to make the capped mRNA. That's the only difference in there. On the other hand, when it is the genome length, that is not needed, and it is unprimed with this end. So these are the two big differences in between these two. One is mRNA synthesis, and another is genome RNA synthesis. So the key concept for these, what we see that again, the plus minus and the double-stranded RNA that we have seen, know the polymerase basic site of the RNA synthesis, and the mRNA synthesis and the genome RNA synthesis and the unique capping mechanism that we just saw, that it steals the caps, and genome RNA synthesis, polyaddition. So these are the main keys. And again, if you think about that, the material could be really intimidating in the sense that there's lots of intricacies can be asked from here. Remember that that is not the purpose of the course, and you're going to see that it is more of a general idea that how things are happening. That's the idea. And your practice quiz will give you a sense of that, what kind of question will be asked in the exams as well that you have seen, and you will feel comfortable. We just went into the little bit more detail compared to that, but other than that, you will see that the questions compared to that what is being presented will not be that difficult. Although understanding of the overall mechanism is very important. So just try to get that why mRNA is being synthesized and why it is switching from the mRNA synthesis to the replication. These are some of the key things that once the mRNAs are synthesized, then it's going to make the nmRNA. This nmRNA is going to translate into N protein. When the N protein is large amount in number, these N protein, as soon as it starts to synthesize, just like this, it's going to cap immediately, encapsulate immediately the RNA that is being synthesized. That is the switch to the replication. So these are some of the very specific things that definitely take a look at that, and very important in there. And here there are some specific one that I just wanted to mention, that the cap length is usually 13 base pair. So the cleavage of the mRNA actually happens. So this is basically the top one is the host mRNA, and this APCP, and this is the mRNA, and the RNA-dependent RNA polymerase cleaves that right here. And then this is used as the primer, and this is added directly to the penultimate base. This is actually a quiz question, and definitely remember that, not the first base, but the second base, which is a C. This is the penultimate base, which is added to this primer. And see that U is not in there. It is right like this, and it starts the synthesizing the gene from there. So this is a very specific question, and sometimes this is in between the lines. So remember that where the primer actually binds for the initiation. So that's pretty much it. So maybe a little bit overwhelming for most of you guys, but still tell me that what do you guys think? Any question? All right, I get so let's do. So how many of you actually do think that it is a little bit overwhelming? Let's see. Little bit overwhelming. Okay. All right. So yeah. So a lot of people are thinking it is a little bit overwhelming. Yeah, I see that as well. But I think that what we are going to see, that as we move through the material, it will get a little bit clearer as well. But this week you will have the quiz as well, and just try to read the material as well, and just wait a couple of minutes. I will just make the quick correction of that one slide, and I'm going to upload that, and I'm going to post a message that please download it now. So what I'm going to see that right here, I just can make it right away itself. Let's see right here. I'm just going to share the desktop. Yeah. So what I'm going to do is that you can see that the TRS, instead of that, I'm going to delete this TRS, and this is all I need. So the TRS is just one of these. So I'm just copying this and pasting this right here. So this one is the TRS. That all you have to know like this. I'm just deleting that. So just remember that it was pointed right here. So this is the one that I'm going to upload right here. So all of these purple that you're seeing is the TRS, and it can make the jump from one TRS to the all the way to the front. Remember that. It will not jump from one TRS to the next one in here, but it all will jump to the front. So all of these actually does have the same coterminal, 3' coterminal, as well as the beginning of this as well. So that's one of the important part that you have. All right, so that's pretty much it, I guess. So I will just going to upload this. Don't worry about that. I'm just going to upload this in a second, so you can download it within 5 minutes. You should be absolutely fine. So that's it about it. Are we good? All right, thanks a lot, folks. Yeah, it is a little bit overwhelming. I will just upload the quiz, and you will see that the quiz questions are not, it is not that easy, but it's not that difficult either. If you just have an overview, nice overview of that, you should not have any difficulty. And this is an introductory virology course. This is not a detailed virology course where we can talk about this chapter, make it a RNA-dependent, RNA-directed synthesis could be just a course by itself, but you do not have to go in that detail. All right, then have a good weekend, and we'll see you next Tuesday. Just only one quiz due. That's it.